Hello, my name is Aysha Hales-Henao and this is the PICO presentation for the World Congress of Soil Science under the theme Soil Carbon from Particle to Planet. Our title is Carbon Sequestration in Restored Heathlands, a Viable Climate Mitigation Tool. Now, just a little bit of context. Soil is a carbon sink and it accounts for a third of the global terrestrial carbon pool. This means it could be a really good resource for carbon sequestration and carbon storage to help combat climate change. Now, our work is in ecological restoration and the restoration of pasture land. Now, agricultural land use change is highly detrimental to the soil, disrupting microbial communities and minimizing the capacity of soil to sequester and store carbon. So, ecological restoration techniques that enhance carbon sequestration have the potential to significantly impact climate change mitigation efforts. In terms of heathland restoration, one factor that's been associated with carbon sequestration is the presence of ericaceous species such as Calluna vulgaris or heather as seen on the right. These species have a higher proportion of lipids and aliphatic biopolymers and are more resistant to decomposition and will accumulate in the heathlands. However, ericaceous species prefer an acidic soil climate, so to restore the agricultural land and encourage the establishment of these species, an acidifying agent needed to be applied. So let's talk about our methods. We had an 18-year-long experiment run on the Isle of Purbeck in Dorset. There were 5 experimental pasture sites, and these each received 2 initial doses of elemental sulphur, and then they were later compared to the native heathland and then the untouched control pasture. We were trying to investigate 2 questions. Primarily, would the carbon content in the newly acidified pasture get back up to the levels of the native heathland? And more holistically, how successful and viable is this technique in restoring agricultural land? Now, moving on to the results. Primarily, the plant community survey. The plants in the control acidified pasture and native heath were analysed and had greater or equal to 50% similarity between the plots. The acidified pasture had a high predominance of the acid and heath species. As previously mentioned, the ericaceous species are important for carbon sequestration, and there was a positive correlation between total carbon and the presence of ericaceous species in the heathland. These species are important as they're held within a secure chemical stabilisation method. Now, an important aspect of this research was understanding the mechanisms of carbon stabilisation and accumulation in heathlands and our acidified pasture. If we look at the graphic up on the right, we can see the 3 mechanisms of organic carbon stabilisation. The 3 mechanisms are active. This is free particulate organic matter, often consisting of litter and small macroaggregates. It takes years to form and it has a low recalcitrance. Then we have slow carbon storage, where carbon is occluded in organic matter, such as microaggregates. This takes decades. And finally, the stable fraction, where carbon is stored in mineralized organic matter. This is the carbon storage that the ericaceous species will eventually fall into. Now, we separated our own fractions in our bulk soil using an organic matter particulate size physical fractionation method that involved a wet sieving technique. On our fractions, we then ran C&N analysis and we identified that the native heathland had a high total carbon and a low proportion of this was active. So it was very stable, lots of carbon, very stable. And we can see that in figures 3 and 4. Now, analysis of the plots that had been treated with elemental sulphur identified that the litter and SMA POM fraction within the acidified pasture underwent the greatest change. However, as indicated in the infographic, the litter and SMA is held within the active carbon storage and is therefore liable and has a low recalcitrance. This is noted in figure 5. Ultimately, as indicated generally in graph 1, after 18 years, the acidified pasture did not have significantly different levels of C or N to the control. This is important as there are very few long-term studies that have been conducted on carbon sequestration in restored heathlands, and knowing that there was no significant difference raises questions regarding policy making and the long-term heathland protection. This long-term protection is even more important as we know that the newly sequestered carbon within the litter and macroaggregate fraction in the newly acidified pasture was in fact active and very likely to decay if it was disrupted. However, carbon stabilisation occurs over centuries, and 18 years of data is really only the very beginning. This research raises lots of questions about climate mitigation efforts and the application of elemental sulphur as a tool for ecological restoration. Thank you for listening to this presentation, and I hope it's raised questions of your own.